Hello all. Again, we continue the second part of the chapter 2, which is about the axon guidance. Okay, and let's just share the screen again, as usual. Here we go. Okay. So we, in a previous lecture, we're talking about the neural crest cells and their development for the particularly the peripheral nervous system. And this part of the lecture, we're talking about how the neurons make a connection with other neurons. Okay, so axon guidance is the one critical event for the formation of the functional nervous system. And this picture is showing the synaptic connections of the one neuron with nearly the thousands of other neurons. So each of these green spots are showing the synapse that which is the physical association with other neurons, basically. So a single neuron can interact with thousands of other neurons, and there's a very ambitious projects launched many years ago, which is called the Human Connectome Project. Okay, Human Connectome Project, or HCP. This project, the aim of this project is to map the entire neuronal pathways in the human brains. So again, we're talking about more than 80 billions of neurons in the human brain. And this project is basically, they develop a very sophisticated techniques first, and then use those techniques to understand what part of the neurons in one part of the brain project into the another part of the brain cells to make the synaptic connections with those neurons. Okay, so it's a very, very complicated technologies involved, not only just the histological technique, but also computational techniques also involved to resolve all these connections, basically. So those are interesting, this kind of project. You can go into the graduate school or medical schools in the neuroscience area, and then you can participate in the project. I don't know how long it's going to take to finish this project, or we ever really be able to finish this project. We don't know, but even if we cannot complete the project completely, but at least we might have a very good idea about these neural connections, and then that allows us to understand the function of the human brains and perhaps the development of the therapeutic strategies to cure some neurodegenerative disease or other neurological diseases. So let's talk about the axon formation first, and then how the axons are guided to specific targets. So first part, I copied from a literature, and this one is showing the developing the neurons. So when the neuron was born first, then it doesn't really have this kind of structure, okay? So it looks like this kind of structure, which is not really polarized structure here, okay? And then the stage number 2, now it gradually generated some kind of the cytoplasmic extensions called the neurites, okay? So these are all immature neurites. And then one of these neurites is determined to be the axon, okay? And the other will be developing into the dendrite. So this axon will be growing and growing and growing to find the appropriate target, okay? So that is the first part of this lecture. So the tip of the growing axon is called the growth cone, okay? Growth cone structure like this. And if you look at this, the terminal of the growing axon over here, then the structure is loaded with cytoskeletons. Okay, so the shaft of the axon, you can see the protein in this shaft is the microtubule cytoskeleton, which is shown in the red. And a little bit of periphery over here, the green area, these are the loaded with the actin filament, okay, fibrous actin or actin filament shown in the green. And if you look at this area a little bit more closely, then you can see the large number of the spikes, okay, large number of spikes is called the microspikes are formed. And the growth cone basically it senses the various chemical signals in the extracellular environment. So it's a large, these are all the extracellular environment over here. It produces various chemical signals that attract or repel the growing axons over here. So depending on the signals they are sensing, growth cones can extend toward the signals or retract. So in other words, they're repelled from that source of the signals. So these micro spikes are growing in that direction or collapsing from these repulsive signals. So it's very dynamically changing depending on the signals that they're receiving that eventually provided the directionality of the axon growth. So this one it's kind of complicated, but if you look a little more closely, then it's not too bad actually. So again, this is the growth cone, and we have the microtubules shaft, and here's the kind of central zone of the growth cone, and a microtubule is kind of now the separated to support the structure of the growth, the microspikes over here. Okay. Now, in this diagram, this growth cone is originally in this shape, but then it's now exposed to repulsive signals on this red area, or the attractive signals on the area shown here in the blue. And of course, as you can imagine, the axon growth will be going this direction where the attractive signal is, and they're repulsed from these repulsive signals. So there are the directionality changes depending on the signals that they are receiving. If they have receiving the repulsive signals, then all these, the microspikes are collapsed, okay, collapsed. And then the new micro spikes, which is called the filopodium, okay, filopodium is formed toward the attractive signals. So the collapse and the formation of the new spikes all depends on the cytoskeletal reorganization, okay? So this is the growing microtubules. And again, the microtubules has the polarities. The plus end means the microtubule is growing that direction, okay? So the minus end, negative end is here, and the plus end is where the microtubules are growing. So growth is dynamically changing depending on the signals that the growth cone is receiving. So here, in response to a repulsive signal, the microtubule is depolymerized at this area. And the area where they are receiving the attractive signals, the microtubule polymerization takes place, so it's elongating. There are two proteins, it's more than two, but two major proteins indicated here. One is called the CLASP, and the other one is APC, okay? The CLASP and APC, both proteins, they are called plus tips, okay? Plus tips, the reason is because these two proteins bind the plus end of the microtubules, basically. Okay. So these two proteins, CLASP and APC, they are in the plus tip, okay, plus end of the microtubules, and basically it stabilizes, and the length together. The plus tips stabilize and lengthen the microtubules, so that allows the formation of the micro spike, which is the filopodium. At the repulsion, there's a disassembly again, the depolymerization of the microtubules. So then it's all the filopodia structure is now collapsed. Okay. So then once they make a decision, move toward the attractive signals, microtubules grow further, along with the actin filament also heavily formed in this area as well. And then they are extending the cytoplasm. That is dragging the structure toward the attractive signals. Okay, and then the C domain moves forward. So this area over here is now further invaded by the microtubules here. So then the new shaft, okay, new axon shaft is formed in that direction, and that allows the entire structure move toward the attractive signals. Okay, so then the gross cone extension in a certain direction requires a bit of a supplies, okay? So what kind of supplies they need for supporting this extension? So one is the, of course it needs the microtubule polymerization. And for the polymerization of the microtubules, we need is the tubulins. And tubulins are the monomers of the microtubules. Okay, so then what you see here is there are quite extensive on-site translation of the tubulin messenger RNA. So the cell body is far away here. Okay, this is the axon, and the cell body is here. If the tubulin is made in the cell body, just like other proteins, those tubulins must be delivered down to the axon terminal, to the gross cone. And that takes quite a long time for this gross cone to respond quite dynamically to these various extracellular environment. So that is not quite desirable. So what you basically see here is all those messenger RNAs actually present in the tip of this, in the gross cone. And then there's a translation is happening in this, axon terminals, okay? So it's called the on-site translation, okay? On-site translation. And that is what's happening to supply, the quick supply of all these tubulins for rapidly growing the microtubules. Okay, second supply they need is the membrane. Okay, so as you know, now as you can see here, if the filopodia is growing this direction quite rapidly, then it needs some the plasma membrane materials to support this membrane growth. Okay, so that plasma membrane component is supplied by the exocytosis of vesicles. So this is the showing the exocytotic events happening in the plasma membrane. So here's a vesicle. Okay, vesicle, of course, is formed by the plasma membrane. Okay, the lipid bilayer. So lipid bilayer, and then it fuses with the plasma membrane. So that provides this much amount of the lipid bilayer to the plasma membrane, so the plasma membrane can be extended. Okay. So basically, this is the histological the evidence showing this local translation. So the dotted line is showing the contour of the gross cone, and the green spots indicate the tubulin messenger RNAs. Okay, so these are tubulin messenger RNAs localized in the gross cone. This is axon shaft over here. APC is the plus tip protein, as I mentioned earlier. Here is the APC protein, one of the TIPS protein, okay, that is associated with the plus end of the microtubules. And that actually also APC is here. APC is facilitating the translation of the tubulin messenger RNA. And this is basically showing that the APC is present in the tip of the growth, the spike over here. And here's the tubulin messenger RNA shown here. Okay, so then we briefly mentioned about this, the guidance cue, okay, recursive signals, subtractive signals. So what kind of signals we are talking about here? Okay, so those signals are collectively called the guidance cues. Okay, there are four major guidance cues illustrated here. So one is ephrins and netrins and slit and semaphorins. And I briefly mentioned about the ephrins and the semaphorins for the neural crest cell migration. Okay, and the other two we'll also talk about these netrins and slit. So these the guidance cues are basically, so these are in the extracellular space over here, and this is the terminal of the axons, and these signals are recognized by specific receptors. So the ephrins recognize the EPH receptor. Netrins binds to the receptor called the DCC or UNC5. The slit receptor is called the ROBO, and semaphorin receptor is the I think it's a neuropilin. I'm not completely sure, but we'll get to that later. Okay, and so once these receptors are activated, basically it regulated the activities of the small GTPase proteins. Okay, these are the three major proteins: RHO, A, RAC1, CDC42,